Okay, so in last lecture we have started about the topic that is histamine, and as it's been many days since we have studied anything about autacoids, let's just revise about histamine a bit, and then we will move forward to the actual points. Okay, so these were the contents of my presentation and the points which we have covered till now. It is about till the release and function of the endogenous histamine. Okay, we have started about the types of histamine receptors, and also we have studied how many types are there, what to which family of receptors these histamine receptors they belong to, and we were supposed to study their locations and the functions or the biological effects are produced when these histamine receptors are occupied by the histamine itself. Okay, so this all we have studied. What is histamine? It is like tissue amine, and this histamine plays a very important role in the allergies as well as in gastric acid secretion. Okay, also recently we came to know that it is also acting as a modulator of neurotransmitter release in the central and peripheral nervous system, and also the competitive antagonist of the H1 receptor. They are used therapeutically in treating allergies, then urticaria, then anaphylactic reactions, then nausea, motion sickness, and insomnia. The antagonist of the H2 receptor, they are effective in reducing the gastric acid secretion. Let us first of all think about this particular thing which we have just studied, that we are talking about antagonist. We are not talking about any of the agonist. Why we are not talking about agonist? Because the agonistic actions of the histamine on its receptor, they are most of the times they are unwanted ones. That means nobody wants to have the allergy or their own. Allergy is itself obnoxious feeling, and nobody wants to have it. So when we talk about the drugs which act on the histamine receptor, we talk about antagonist. We are not even we are going to consider about the agonistic action. Histamine as a autocoid, we are going to see what effect it produces when it acts on its receptor, but we are not going to consider any drug example which acts as an agonist on the histaminergic receptors. Okay, so distribution, we have seen mostly histamine present in the mast cells as well as it is present in the basophils. Okay, so biosynthesis, it is obtained from the amino acid that is histidine in presence of an enzyme that is L-histidine decarboxylase. So it is type of decarboxylation reaction. Okay, so storage and metabolism. Storage, it is stored in mast cell and the basophils, and the metabolism, it can happen at slow rate in this particular storage site, but non mast cell storage site shows higher turnover rate. Okay, so there is a particular reason to it why the turnover rate is rapid in the non-mast cell site. The reason is that histamine which is produced over there, it is being released continuously. It is not stored in those particular sites. Okay, and this histamine which is released, which has acted upon its receptor, it is rapidly metabolized by either ring methylation, which is catalyzed by an enzyme that is histamine N-methyl transferase, or oxidative deamination, which is catalyzed by the diamine oxidase. The metabolites, they are eliminated in urine. So if we do the urine analysis, we will find the metabolites over there. Okay, so this is the chart, or these are the two fates of the histamine metabolism. One fate is in presence of N-methyl transferase and another is in presence of diamine oxides. Okay, so what is the end result of N-methyl transferase is that we get N-methyl imidazole acetic acid, and in presence of diamine oxides we get imidazole acetic acid riboside. Okay, so release and function of endogenous enzyme. So it is released from the storage granules as a result of interaction of antigen with the IgE antibody on the mast cell surface. So antigen antibody reaction takes place on the mast cell surface, and thus the histamine is released from its storage granules. So what function it plays once it gets released? It causes immediate hypersensitivity. That is the type 1 hypersensitivity reaction. It is because of the release of histamine from the mast cell, and also it plays very important role in the allergic responses. So the histamine on the bronchial smooth muscle and the blood vessel, they account for many of the symptoms of the allergic response. So whatever allergy we see, that is because of the action of histamine on the blood vessels as well as in the bronchial smooth muscles. Okay, so the histamine, it also acts as leukocyte chemoattractant, and it also plays a major role in regulating the gastric acid secretion, and it modulates neurotransmitter release. In addition, some drugs, they act directly on mast cells to release the histamine, and thus they cause the untoward effect or unwanted effects. Okay, so the types of receptor, we have four types of receptor, that is H1, H2, H3 and H4. The H1 receptor, it is all of these receptors, they belong to the family that is G protein coupled receptors, and the H1 receptor, it is internally coupled to the GQ G protein, and thus it activates the IP3 DAC pathway. Okay, and thus calcium, uh, it has many sequels or many effects therefore. So it activates the PKC, then calcium calmodulin depending enzymes like eNOS and various protein kinases, and also it leads to activation of PLA2. The H2 receptor, it is linked internally to the GS protein, G protein, and thus it acts with the adenylyl cyclase CAM pathway. Okay, so H3 and H4 receptor, they are internally coupled to the G protein, and thus they inhibit the adenylyl cyclase, and thus they decrease the intracellular CAM. Okay, so the activation of this H3 receptor, they can also activate the MAP kinase enzyme, and also they inhibit the sodium hydrogen exchanger. The activation of H4 receptor, it can mobilize the stored calcium. The H3 and H4 receptor, they have about 1,000-fold higher affinity for the histamine, that is in low nanomolar range, and when we compare it with that of the H1 and H2 receptor, it is so much so that. Okay, so activation of a H1 receptor on vascular endothelium, it stimulates the eNOS, and thus it causes production of nitric oxide, and this nitric oxide thus diffuses to nearby tissue, muscle cells, to increase the cyclic GMP, and thus it causes the relaxation. Stimulation of the H1 receptor on smooth muscle, it will mobilize the calcium, and thus it causes the contraction. And whereas activation of H2 receptor on the same smooth muscle will link via GS, that is uh, G protein, to enhance the cyclic AMP accumulation, activation of PKA, and then it will lead to the relaxation. So if we see the actions the histamine produces on a same smooth muscle via two different receptors are the two exactly opposite actions. When it binds to the H1 receptor, it will cause the contraction, and this contraction, it is mediated by the calcium. But what about the H2 action? When the histamine binds to the H2 receptor, it will cause the relaxation of that same smooth muscle. but this relaxation is carried out by the cyclic AMP. So you should be able to correlate. This is not only limited to the histamine, but it is common thing for all. Wherever the intracellular mechanism is the IP3 DAC pathway, then it will lead to the contraction of that particular tissue. And in case of, if the intracellular mechanism is associated with increase in the intracellular camp, then it will lead to the relaxation. It is a common theme. As you will read about the actions, pharmacological actions of many of the substances, you will find this a common theme in them. Okay. So next is, let's see about H1 and H2 receptors. So H1 and H2 receptors, they are distributed widely in the periphery and in the CNS, and their activation by histamine, it can exert local or the widespread effects. So it depends where histamine it is getting released. If it is getting released locally, then we will get only localized, like focused responses. But if it is acting on the CNS, then we can expect widespread effects. So it contracts many smooth muscles such as those of the bronchi and the gut, which are nothing but the H1 mediated actions. But they markedly relax others, which include those in the small blood vessels. So the histamine also is a potent stimulus of gastric acid secretion, and this action of the histamine, it is H2 receptor mediated action. H1 receptor has no any effect over there. The reason to it is that H1 receptor are not to be found in the gastric mucosa. Only H2 receptors are present. That's why this action is H2 mediated, and the gastric acid secretion, it increases when histamine binds to the H2 receptor. In the CNS, H1 activation, it inhibits appetite and it increases the wakefulness. Okay, so the vascular dilation, it is mediated by both H1 and H2 receptor stimulation. Now this one, we are very confirm about the H2 receptor, but the actions of H1 receptor, they are quite confusing. Why they are confusing? The reason is that H1 receptor, if you see, it is internally associated with the IP3 DAC pathway. And what I just said, that this IP3 DAC pathway, when it gets activated, it leads to the contraction. Then why there is vascular dilation? If you remember, I have also mentioned in last slide that activation of H1 receptor on vascular endothelium, it stimulates eNOS and it produces the nitric oxide. So if nitric oxide is being produced, and as we know, nitric oxide is a potent vasodilator, that's why the H1 receptor in this condition causes the dilation, which is exactly opposite to its nature. Okay, but still it happens. Okay, so H3 receptors, they are expressed mainly in the CNS, and especially they are present in basal ganglia, hippocampus and the cortex. The presynaptic H3 receptor, they function as autoreceptors on histaminergic neurons, and thus they inhibit the histamine release and modulate the release of other neurotransmitters. The H3 agonists, they promote the sleep, and H3 antagonists, they promote the wakefulness. So understand these, the H3 receptor, they are associated with the sleep and wakefulness cycle. Okay, so agonist, if they are promoting the sleep, that means the H3 receptors, when they are stimulated by the histamine, they will cause us to sleep. Otherwise the antagonist over there, they will cause the wakefulness. Okay, so H4 receptors, they are primarily found in eosinophils, dendritic cells, mast cells, monocytes, basophils, and the T cells, and but have also been detected in the GI tract, dermal fibroblasts, CNS, and primary sensory afferent neurons. So if you see the H4 receptors, they are found in a variety of locations. Okay. So activation of H4 receptor, it has been associated with induction of cellular shape change, chemotaxis, secretion of cytokines, and the upregulation of the adhesion molecules. And it suggests that the H4 antagonists, they may be useful inhibitors of allergic and inflammatory responses. Okay. So although specific H3 and H4 receptor antagonists, they have been developed, none of these agents has yet been FDA approved for clinical use. Okay, so based on the functions of the H3 receptors in the CNS, H3 antagonists, they have potential in treatment of sleeping disorders, then ADHD, epilepsy, cognitive impairment, schizophrenia, obesity, neuropathic pain, and the Alzheimer's disease. Okay, so these are all probable target diseases which we can treat with the help of H3 antagonists. But one thing we have to remember, whatever H3 antagonists have been introduced, or they are still in the developmental stages. None of the drug is approved for the clinical use. They are still in clinical trials. Okay, so because of the unique localization and function of the H4 receptors, H4 antagonists, they are promising candidates to treat inflammatory conditions such as allergic rhinitis, asthma, rheumatoid arthritis, and possibly pruritus and the neuropathic pain. Okay, so earlier conditions, we are quite sure that we can use the H4 antagonist, but we are not very sure that we can use this in pruritus and the neuropathic pain. Okay, so next you can see the chart. It is like comparative chart of all the four receptors. The representative agonist and the representative antagonist, they are given. Why it is said as the representative agonist and antagonist? Because there are many drugs. Okay, not all the drugs we have mentioned over here. Okay, so we are only going to focus on the antagonist of the H1 and H2 receptor. We are not going to see any of the drugs that act on the H3 and H4 receptor. May it be the agonist, may it be the antagonist. Okay, so the reason to is that there is no drug which is FDA approved, and we will be considering only the drugs which are there in the market since ages. Okay, and the H1 and H2 blockers, they are there in the market like last 50 years. Okay, so let's start with the pharmacological actions of the histamine, and only this much we will study today. Other part we will study in next lecture. Okay, so histamine, it produces pharmacological actions in following systems, tissues, and organs. Okay, so which are these systems? Cardiovascular system, extravascular smooth muscle, then peripheral nerve endings, and we are going to see specific thing, that is histamine shock. Okay, so why it produces pharmacological actions only in these systems, not all other system? The reason is quite clear, is that because of presence of the receptors in these particular systems. Okay, if histamine receptors were to be found all over the body, then it would have produced the effects all over the body, but it is not the case. The histamine receptors are located at particular locations, so in those particular locations only the histamine will produce its actions. Okay, So let's see the first system onto which histamine produces the effects. So in cardiovascular system, histamine dilates resistant vessels. So you should understand which are the resistance vessels and which are the capacitance vessels. Okay, to understand this, okay, I don't require to tell you which are the resistant vessels because you have already studied this. Okay, so it increases the capillary permeability and thus it lowers the systemic blood pressure. In some vascular beds, histamine it constricts the veins, contributing to the extravasation of the fluid, and thus it leads to the edema formation upstream in capillaries and post-capillary venules. Okay, so vasodilation is the another effect of the histamine on the cardiovascular system. So vasodilation is the most important vascular effect of histamine in humans, and it can result from activation of either H1 or H2 receptor. We have just seen H1 receptor, when it gets activated it causes formation of nitric oxide, and the H2 receptor when get activated it causes increase in the intracellular CAM formation. Okay, so this is what is said in next two sentences. Same. So as a result, H1 antagonists they effectively counter small dilator responses to low concentrations of histamine, but blunt only initial phase of larger responses to higher concentrations of amine. Okay, so what what is the meaning of this sentence? The last sentence is that H1 receptor antagonists they are useful only in those cases when the histamine the presence of histamine in a particular area is very small, that very little amount of histamine is released. But in if the histamine concentration if it is large, then in that case the H1 receptor antagonists they produce very little effect. Okay, so next is increased capillary permeability. Increased capillary permeability means what? The capillary spaces, the gaps between the adjacent cells of the capillary, it gets increased because of actions of the histamine. If you remember the chapter that is inflammation and tissue repair, in that we have studied about various mechanisms that that are responsible for the increase in the permeability. If you remember the acute inflammation part, the hemodynamic changes happen, then vascular events they take place. In vascular events there is change in the permeability, and one of the change in the permeability is brought about by the presence of the histamine. So what happens in this case? Histamine's effect on the small vessels, it results in efflux of plasma protein and fluid into the extracellular spaces and an increase in the lymph flow, and thus it causes edema. H1 receptor activation on the endothelial cells is the major mediator of this particular response. Okay, and it leads to the GQ mediated activation of RhoA and the ROCK, and thus it stimulates the contractile machinery of the cells, and thus it disrupts the interendothelial junctions. So what we mean to say is that when histamine acts on H1 receptor, the gaps between the adjacent cells that form the lining of the capillary endothelium, it increases, and as the gap increases, whatever contents are there in that particular small blood vessel they come out, Usually they remain in that particular blood vessel only, but due to the action of histamine, these contents they come out. Okay, so these gaps between the endothelial cells also may permit passage of the circulating cells, which include the RBCs also, recruited to tissues during the mast cell response. So the recruitment of circulating leukocytes is enhanced by the H1 receptor mediated expression of adhesion molecules. Example is the P-selectins on the endothelial cells. Okay, so these leukocytes we have studied that rolling adhesion and all diapedesis, that process, immigration process, in that this particular H1 receptor they actually cause increase in expression of the adhesion molecule. Which adhesion molecule? That is P selectin. And as the adhesion molecule expression is increased, the adhesion phase of that particular leukocyte migration process, it is facilitated by the histamine when it acts on the H1 receptor, and by that the leukocyte they come in contact with the endothelial cells, and at appropriate location these leukocytes they exit the blood vessel and enter the tissue spaces, and this is made possible because of the actions of the histamine. In this case, if you see the in inflammation, in my immigration of the leukocyte, the histamine plays very important role. First thing it does that it increases the capillary spaces, and the second thing is that it increases the expression of the adhesion molecules, and thereby it facilitates the emigration of the leukocyte outside the blood vessel. Okay, so triple response of Lewis. So this we have already seen what is triple response. So I won't take much of a time. That is red spot, will and flare. Why this happens? When if you poke your forehand with a sharp pointy thing, then the histamine which is getting released over there, it will produce a characteristic response, and this response it is called as the triple response of Lewis. Okay, so reddening it is because it will happen within the few seconds and maximal at about 1 minute. So flare or red flush it extends about 1 centimeter beyond the original red spot, and it develops more slowly, and the wheal or the swelling it is discernible in 1 to 2 minutes at the injection site. Okay, so the effects on the heart. Histamine affects both cardiac contractility and electrical events directly. So increases force of contraction of both the atrial and ventricular muscles by promoting influx of the calcium, and it speeds the heart rate by hastening the diastolic depolarization in the SA node. Okay, hastening means it is like moving forward in a fast pace. Okay, so not giving the time for that SA node to depolarize, completely keeping the SA node in the sorry, not in repolarized state, keeping the SA node in depolarized state only continuously because of the influx of calcium. So it also directly slows the AV conduction to increase automaticity, and in high doses this can elicit the arrhythmias. Okay, so the slowed AV conduction we have seen this in antiarrhythmic drugs also. So up till a certain level the AV conduction, if we slow it, then it will actually rectify the arrhythmias, but if it is slowed beyond limit, it can lead to the arrhythmias. Okay, slowed AV conduction, it involves mainly the H1 receptor, while the other effects are largely attributable to the H2 receptors and the cyclic AMP accumulation. So the direct cardiac effects of histamine given intravenously are overshadowed by baroreceptor reflexes stimulated by reduced blood pressure. Okay, so if we try to elicit this response by giving the histamine, we won't be able to get these responses. The reason is that our baroreceptor reflex is too strong to overtake by the histamine. Okay, so extravascular smooth muscles, histamine directly contracts, or more rarely it relaxes various extravascular smooth muscles. So contraction is due to activation of H1 receptor, simple IP3 DAC pathway, which leads to the calcium release. Okay, and relaxation is mainly due to the activation of H2 receptors. Although spasmogenic influence of H1 receptor is dominant in human bronchial muscles, the H2 receptors with dilator function are also present. Why the spasmogenic influence of H1 receptor is dominant? It is because solely because of the number of H1 receptors is more in the bronchial smooth muscle when we compare it to that of the H2 receptors. So the peripheral nerve endings, what effect histamine has on over this? That histamine stimulates various nerve endings and thus causing sensory effects. In the epidermis, it causes itch. In the dermis, it evokes pain, and sometimes it is accompanied by itching. So stimulant actions on nerve endings, which include autonomic afferents and efferents, it contribute to the flare component of the triple response and to the indirect effects of the histamine on the bronchi and the other organs. Okay, so histamine shock. What is histamine shock? So this will be the last slide, I guess. So histamine given in large doses or released during the systemic anaphylaxis, it causes a profound and progressive fall in blood pressure. Okay, so as the small blood vessels they dilate, they trap large amounts of blood, their permeability increases, and the plasma escapes from the circulation. These effects, they resembling the surgical or traumatic shock, they diminish effective blood volume as most of the plasma leave the blood vessel and enter the tissue spaces. They reduce the venous return and also cause greatly lower the cardiac output. So similar condition we have already studied in the congestive cardiac failure. Okay, so that was all about the histamine shock. Okay, so this histamine shock we can see only if we give the histamine in very large doses or in very large amounts. The histamine is released during the anaphylactic shock. Otherwise, there is very little chances of we getting any histamine shock. Okay, so this histamine receptor agonist and histamine receptor antagonist, this part we will be studying in next lecture. For this lecture, we are good till this particular last point, that is histamine shock. That's all from my side. Thank you.